Bueno, muy buenas tardes tengan todos. Espero que se encuentren muy bien. Este es parte del coloquio del Instituto de Física de la UNAM. Y bueno, en esta ocasión tenemos al profesor James Glazier de la Universidad de Indiana, quien pues nos va a hablar un poco más de él, el doctor Gerardo García Naumas. Antes que nada, les comento que, bueno, estamos transmitiendo por YouTube y por lo tanto, aquellos que, bueno, que no quieran aparecer y demás, se pueden unir en YouTube o apagar su cámara. Finalmente, pues yo quisiera hacer un agradecimiento público a Grisel Méndez, quien nos apoya muy activamente a contactar, a formar toda esta parte de los coloquios. Nos ha apoyado ya a lo largo de más de un año y quisiera yo hacer este agradecimiento público. Y adelante, muchas gracias por estar aquí, profesor Glazier. Y Gerardo, adelante, por favor.
Muchas gracias, Cecilia. First of all, I would like to thank Professor Glaser for accepting the invitation. It is really a pleasure for us, especially because this kind of topic is very important for us. We want to really develop in this direction some part of the institute. So let me read just a few words about the CV of Professor Glaser. He received his BA in Physics and Mathematics from Harvard University and his Master in Science and PhD in Physics from the University of Chicago. He has held faculty positions in physics at the University of Notre Dame and Indiana University at Bloomington, and visiting positions at the University of California, Santa Barbara, the University of Western Australia, the University of Grenoble in France, and Tohoku University in Japan. Professor Glazier's research focused on applying physics-based computer simulation to understand embryonic development, developmental and chronic toxicity, and developmental infectious diseases. He also applies this simulation to develop and optimize diseases treatments. He leads the collaborative development of the open source code Computational 3D multi-scale modeling environment. In 2016, he became one of the founding members of Indiana University's Department of Intelligent System Engineering, the first engineering program at the Indiana University at Bloomington, which aims to apply advanced computer techniques to understand and control complex neutral and engineering emerging systems. He's a fellow of the American Physical Society and the Institute of Physics, London, and has served as chair of the Division of Biological Physics of the American Physical Society. He currently co-leads the IMAG-MSM working group on multi-scale modeling and viral pandemics. So thank you very much for accepting the invitation.
Well, thank you very much for having me and spending an hour with me this afternoon. I know some of you have heard parts of this talk before the last time I was at UNAM, so I appreciate your patience if I cover some of the same material again. I'd like to start by acknowledging the many collaborators whose work I will be talking about today. I may have left some people out. I'd like to say that one of the people on the list is Javier Toledo, and he was a student at UNAM of Gerardo's, and came and did a postdoc with me, then moved on to a fellowship at Vancouver and UBC, and we continue to collaborate. So we have collaboration from my last trip to UNAM that was very productive. So without going through more, I'm going to be talking today about some things that may be rather far in some ways from the physics, core physics ideas. We're thinking really these days about improving our biological understanding and our biomedical understanding. And one of the things that we face in modern medicine is that while engineered systems like airplanes or computers are designed, medical interventions generally are not designed. They don't use computing to design the interventions. That's not that computing isn't important in biomedicine and biological research, but the idea of being able to design biological outcomes is still something that's not possible. Another thing that happens with medicine that we see very much today is that we can't really predict what's going to happen to individuals. Most medical care is developed around the idea of doing what's best for a population as a whole, but that often is not optimal for any given individual. And in particular, in the case of COVID, we can't predict what's going to happen individually to people. When people get a vaccination, you can't predict how long the vaccine will last. When people get sick, you can't predict how sick they're going to be, except in very coarse-grained ways. We're quite a bit away from being able to do something about that, but I think it's worth thinking about a long-term goal of designing simulations that are what are called digital twins, ways of actually predicting the health outcomes of particular individuals rather than populations. And we wrote a little editorial in Science a few weeks ago addressing some of these issues. And I think physicists have a lot to contribute in all aspects of thinking about the digital twin problem, everything from data analysis, modeling, to designing the sensors that would be needed to make digital twins a reality. How do you monitor the state of health of an individual? We don't really have ability to do that yet. And so I think there are a limitless, if they're graduate students on this call, I think there are really a limitless number of problems in the area of biomedicine and medical modeling that could be very appealing to a student. You could easily have many careers worth of research topics. If we're going to predict how health state changes, we have to be able to understand it. And if we look at the life cycle of an organism, you start with a fertilized egg, and then there is the process of development. And that is how I became interested in these kinds of problems, because I worked in pattern formation, in fluid dynamic pattern formation, as it happened. Then something that is maybe a little less obvious, because in physics we're used to things that are equilibrium. In biology, you're never in equilibrium unless you're dead, which is how do you maintain an organism? That's called homeostasis. And that's actually quite a difficult problem. The way you repair an airplane is different from the way you build an airplane. But in biological systems, the way things are built and repaired are the same. And the way developmental mechanisms are repurposed to maintain organisms is fascinating and complicated and not completely understood. Many of the things that we call developmental diseases, like cancer, as the classic example, are a result of the failure of those normal repair mechanisms to do the thing that would keep the organism alive. And typically, it's not that those mechanisms aren't working, it's that the control of those mechanisms has changed in a particular way. We also now are interested in infectious diseases. How do pathogens and host interact? And the immune system in particular is central to almost all maintenance of health and almost all diseases. And while we know the components of the immune system, how the immune system works is still poorly understood. Ideally, we'd also like to be able to design therapies to control biological processes and repair them. Bioengineering and tissue engineering are aspects of that. And something that perhaps is not going to be talked about so much today, but it's also quite interesting, is toxicology. If we breathe polluted air or swallow toxicants, poisons, how do they affect our body, and what determines whether our body recovers from those insults or we develop serious problems? All of these are problems that include a physical component and also signaling components, primarily chemical signaling, but also electrical and other kinds of signaling. If I'm talking to a biological audience, many biologists might ask the question, why do we need modeling at all? One of the questions that we might run into is a situation where in physics, we tend to look for the simplest explanation. And we assume that if we have one explanation that is simple, that reproduces our desired results, that that's sufficient. Biology doesn't work that way. Occam's razor doesn't work in biology so much. And so very often we will see situations like this, where molecule A increases the amount of molecule B and molecule C, but molecule B increases the amount of D and C decreases it. And if you ask, if I increase the amount of A, does D increase or decrease? I can't, from looking at the diagram, determine that. I have to know the mathematical details of those relationships. Another kind of question would be, if I put a cell on a substrate and change the rigidity of the substrate, does the cell move faster or slower? There's no simple way of understanding that without doing a biophysical model of how the cell moves. And so this kind of modeling that we talk about, quantitative modeling, it's always going to be phenomenological. Nothing is first principles here. There's a lot of feedback. There is a lot of emergent behavior. And we're often asking these questions in contexts where you cannot simply infer the outcome from looking at the inputs. Physicists like to model things that are homogeneous. We've all learned renormalization group when we do statistical mechanics. One problem in biology is that things are extremely inhomogeneous. And so, unfortunately, many of the fancy mathematical techniques that we learn in solid-state physics, or statistical physics, for example, don't work very well in biology because we don't have homogeneity to fall back on. However, there are particular scales of problem, and there are structures at different scales, typically what's going on inside of cells. And if we zoom into a cell, the organization of cells is extremely complex, and there's a whole world of biophysics trying to understand that. What cells do outside the cell behaviors, how cells organize to form tissues, and then higher level organization as well. For example, the transport of blood in the body or the transport of lymph in the body. And so we have different structures and different principles of organization at different scales. All of these interact together, and those sets of interactions can be modeled in different ways and also function in different ways. And that means we deal with problems that have very large ranges of time scale and length scale. What we call virtual tissues are an attempt to deal computationally with some of that complexity. They make a lot of simplifying assumptions. Typically, we model what is happening inside of individual cells. Those could be signaling networks, metabolic networks, or regulatory networks that turn genes on and off. We model the organization of cells locally within tissues. What are the cells doing? What forces do they generate? What cells are created or destroyed? How do they move? We model the extracellular environment of the cells. What's diffusing in the environment? Is there active transport in the environment, and so on? And then typically at larger scales, we coarse grain things, and we use often what are called PBPK, physiologically based pharmacokinetic models, which are models of transport, mainly in the blood. And those are used especially in biomedical applications. These days, any drug that's used medically will have been modeled so that you understand when you swallow a pill, how much of that drug is available at what time for how long in each tissue in your body. So virtual tissue simulation typically focuses on the scales of control networks, cell behaviors, and then tissue architecture. This is an old simulation showing the formation of the somites in a chick embryo. In somatogenesis, an initially contiguous band of tissue, the presemitic mesoderm, has a symmetry breaking, which leads to the formation of these spherical aggregates. And those spherical aggregates will do a lot of other things. Eventually, they'll organize to form the vertebra in the body. The other thing we do is we develop software. CompuCell 3D has been something we've worked on now for almost 20 years. It's an open source software package that's designed to build models of this kind, these complex models. And we've spent a lot of effort trying to make the modeling as simple as possible. It's not perfect by any means, but it allows you to build in rather compact form very complex models. And all of the simulations I'll show today were built on the same platform. You can download it. It's an open source package. You can download it for Windows, Mac, or Linux. If you don't want to run it locally, we have a server system that we use provided by Purdue University and University of California, San Diego, called NanoHub. And you can run CompuCell on their servers as a web application if you're interested. All of these kinds of models that I'm going to show today came out of work that I started back when I was a graduate student. And fundamentally, like everything or many things in physics, everything starts out with the Ising model. So we start out with the Ising model for phase transitions in a material. And then later on, as a student, I worked on extending the Ising model to understand the dynamics of coarsening in metal grains and in soap froths. And then later on, we extended this to try to understand biological processes. You could say, how does the Ising model have any connection to a chicken? It's not obvious, perhaps, to begin with. And the basic question is, all of these systems, magnetization, soap froths, and living tissues, are soft materials. They all have domain structure. They all have stochastic fluctuations, and they all lead to temporally evolving patterns. And when we're going to build a representation of a tissue, we have to have some idea of the biophysics that we're going to be representing. But the key idea we're going to work from is the idea that cells are an organizing principle in biology. We're going to treat cells, individual cells, as active agents. So if you work in active matter, this will be very familiar. And what we have to do then is build models around the behaviors of cells. And those behaviors of cells typically are going to be organized in terms of energies of interaction, energies that move cells, energies that change their state, And those interaction energies are then going to be the description of the underlying physics or biophysics of the tissue. So if we're going to be describing a biological pattern, we need to be able to describe the configuration of the agents or objects in the problem. We have to be able to describe the forces that act on them, and we need to have a dynamics. Typically, we're in an overdamped regime rather than a Newtonian regime when we're working at small scales, and fluctuations may also be large, and so the dynamics may not be Newton's law. There are many different computational ways of representing cells to simulate them. You can have cellular automata where the cells are points. You can have center models where the cells behave as points with spherical interaction potentials. This would be like a molecular dynamics simulation in chemistry. You can cluster those points together to make blobs, what are called subcellular element models. There are a variety of more mechanical models of cell interactions. In our case, we use a methodology that I developed a long time ago, which is a lattice-based method derived from the Ising model and our Potts models of foam. If we reach back to our memory of the Ising model, we have a lattice. At each lattice site, we have an index, +1 or -1. We write a Hamiltonian, which depends on whether the neighboring lattice sites are co-aligned or not. And then we can add a bias term for an external magnetization. If the interaction energies have a positive prefactor, then you're in a ferromagnet. If they're negative prefactor, they're in a ferromagnet. If they're positive prefactor, you're in an antiferromagnet. You need a dynamics on top of that. The classic physics dynamics are not the ones you use in biology because you don't have detailed balance in biology. And so we have a simple algorithm where you pick a lattice site. You imagine the lattice site overwriting its neighbor. You calculate the change in energy when you do that. And then you accept the change with the Boltzmann probability. That leads to stochastic fluctuations in the pattern. Exactly what the temperature scale means, the energy scale of the problem means, is something that has to be thought about a bit if you want to make things work. The big advantage of this methodology is that if you write not a Hamiltonian in this case, but rather a generalized energy, pseudo-Hamiltonian, and you evolve under that stochastic algorithm, you discover that the pattern evolves with a velocity that's proportional to the gradient of the Hamiltonian. And that's a very useful concept because it gives you overdamped dynamics. The other advantage is that whatever your pseudo-Hamiltonian is, the pattern will solve it. And that means that you can write an arbitrary sum of behaviors or interactions, and this algorithm solves them automatically, and it's totally numerically stable, which not all numerical methodologies are. If you do this just by increasing the number of ground states of your simulation, you get something called a Potts model. Ising model has 2 states, +1, -1. The Potts model has an arbitrary number of identical degenerate states. And if you run the simulation there, let me try that again. You start out with small domains, you get a ferromagnetic interaction leading to domain growth. Then you get something that looks like this. And that goes back, goodness, more than 30 years. And that's where this kind of modeling got started. To go from there to biology, you have to introduce a lot of other energy concepts and also some concepts that run parallel. The idea of, for example, chemical fields and diffusion, interaction between external chemical concentrations in cells. And so you have to give cells the properties that they would have in a biological context. volume, polarity, surface area. If you have connections between cells that represent things like springs or links, you have to give them mechanical properties and physical properties. Chemical fields have to have diffusion and decay properties, transport properties. And because biology has control, you have to be able to model the control that's changing the way the cells respond to their environment. And those are typically done with networks, either of ordinary differential equations or stochastic probabilistic models. A simple example of this kind of extension would be to say that you have two cells that are in contact with each other, and the contact energy per unit length of the boundary between cells depends on the kind of cell on either side. In reality, the cells have particular adhesion molecules on their surface, and those adhesion molecules determine how much the cells stick or don't stick to each other. You need to constrain the volume because you don't want your cells growing the way the soap bubbles do. And you do that using a quadratic constraint. You're basically imposing the ideal gas law on your cells. And that's actually a standard thing you can do. And if you put that together, you get an old-fashioned simulation, which we did back in 1992, called cell sorting, where in this case, the adhesion energy between the blue cells is lower than between the green. And so the blue cells phase separate to form a coherent domain surrounded by green cells. You can use these kinds of things to do more elaborate simulations. In this case, the target volume for each cell evolves in time. It grows. We have a separate algorithm, an event-based algorithm that causes cells to divide, and this allows us then to simulate growing and dividing cells. Chemical fields, you've already seen this picture. In this case, the chemical field is the color in the bar in the middle. We have the ability for cells to secrete and absorb from the outside. There's diffusion of the chemical, and usually a decay term as well. Cells can respond to external chemicals. Chemotaxis is when a cell moves up or down a gradient of an external chemical. That has the mathematical form of a chemical potential. And so that's another kind of term that would describe a particular behavior of cells. You can do fancier things. You can take your cells and you can make them large and put compartments in them to represent structures like the nucleus of the cell. You can have chemicals inside the cell that interact with the cell membrane in particular ways. That's an example here. Here's a simulation of a crawling cell. You'll see this simulation again, I think, in another slide, where you have a simulated field within the cell to represent actomyosin behavior. So you can then take these simple ideas and generalize them. This is an example. All these are from a PhD of a student of mine from quite a few years ago, who's now a professor at the University of North Carolina, who worked a lot on how to build subcellular models. This is a model of signaling within the cells, where the contact between the cells leads to polarization of the cells, and that contact propagates through the tissue. You can also introduce the idea of links, that is essentially springs that connect the centers of mass of these cells. This is a simulation of a process called convergent extension in 2 and 3 dimensions, where we're superimposing on the cells themselves this kind of longer range, more persistent interaction. These are what are called bonded interactions. This is another one of Giulio's very pretty pictures. I wanted to show it mainly because it looks a lot like ferromagnetic transition, except in this case, the ferromagnetic orientation of the cells is not at the single pixel level, the way it would be in your Ising model, but rather is coming about because of the interaction at the biochemical interaction at the surfaces of the cell. This is another movie of that beautiful simulation of Gilberto Tomas in Brazil, UFERGS in Brazil, understanding the statistical behavior of cell crawling. The way cells crawl on a substrate has rather interesting statistical properties. It's a biased random walk with very particular statistics that he studied in a great deal of detail. Okay, another example that we might look at would be a blood vessel development. This is something we worked on many years ago and have used a lot, which is that during the early formation of blood vessels, this is done in vitro. This is in a dish. These are human umbilical vein endothelial cells. They self-organize to form this network structure. And we were very interested in trying to understand the mechanisms which would lead to this kind of network organization. In this case, the interaction is through both adhesion, chemical secretion, and the movement of cells up and down chemical gradients. You can take a simulation like that of vasculature, combine it with a simulation of growing cells and the secretion of oxygen and nutrient fields, and you can use it to build a simulation of a tumor growing in the presence of blood vessels and ask the question about what the different regimes of growth are of that growing tumor and how they depend on, for example, whether the blood vessels begin to proliferate in response to the tumor or not. So those are some basic ideas of this kind of modeling. I'd now like to very briefly give you some examples of slightly more applied, but also biomedically perhaps more interesting problems. And we may have to skip through some of these pretty quickly. It's just to give you a flavor for the different kinds of problems you can solve using these kinds of approaches. And again, I'll remind you, all of the simulations I'm showing use the same modeling platform. However different they look, they're all built on the same fundamental concepts. One of the things that we worked at a lot early on was how blood vessels invade the retina. In particular, in older people, the blood vessels that live behind the retina can cross the barrier between the eye and the supporting choriocapillaris, the blood vessels behind the retina. In this case, in purple, we have the blood vessels. This gray layer is a membrane that normally keeps the blood vessels out. The blood vessels are invading the retina. Once they're in the retina, they proliferate and they begin to destroy the mechanical integrity of the retina. This was a simulation we actually validated pretty carefully. And so the actual simulated time scale is correct from a biomedical point of view. The proliferation of the blood vessels can destroy the physical integrity of the retina and lead to breakdown. And that can lead to blindness over a period of about a year. This typically happens in the elderly, but it can also happen in younger people. And we were very interested in trying to understand the different mechanisms which could lead to different patterns of damage in the retina. Weak RPE stands for reduction in the barrier function of the black layer of cells at the back of the retina, the pigmented retinal cells. That, in fact, doesn't lead to the kind of invasion so much that we saw. Weak RPE-POS, that's up here, is adhesion between the barrier cells and the photoreceptors. That happens when you have inflammation. It also can happen if you have trauma. And so if you're a young person and you're in a motorcycle accident, for example, that's a common problem. That can lead to failure. And then you can also have a situation in where the pigmented cells stop sticking to the substrate that they live on as effectively. And that actually seems to be the primary cause in this case of this particular disease. So in a sense, this complicated disease is actually a change of adhesion the physics of adhesion of the structure. We took those ideas and applied them to trying to understand diabetic retinopathy. This is now a disease that affects the blood vessels not in the back of the retina, but the front of the retina. The blood vessels that normally you see if you do a retinal scan. In diabetics, you see those blood vessels become tortuous. They become squiggly. You see leakage from those blood vessels. You see disappearance of blood vessels. And again, that can lead to blindness over periods of weeks to months. The current treatments for that are using drugs that inhibit blood vessel growth. And it's also that people go in with lasers and actually destroy patches of the retina. This is what you do, essentially forest firefighting, where you cut trees down to prevent the fire from spreading. The problem with both of these treatments is that they're damaging to the retina. And so the treatment itself is causing problems. And we were trying to understand the feedback mechanism that leads to successive failures. Essentially, how does the fire propagate in the forest? And based on that, we were trying to design more effective treatments where you would only cut the trees down. That is, you'd only destroy the photoreceptors in regions where there was high risk of fire. And so this was work that we did to try to design optimal treatments for this disease. Another thing that we've worked on quite a bit, which wound up being a rather nice physical problem, was a relatively rare disease called polycystic kidney disease. It's a genetic disease which causes the kidneys to continue to grow and form spherical cysts. In this case, the genetic cause of the disease was well understood, but how that gene perturbation translated into cysts was not understood. In this case, the hypothesis was that there was an adhesion defect. That wasn't the standard hypothesis, but our collaborator, the surgeon who came to us with the problem, Bob Bacalhau, had this idea that that was what was going on. And we were able then to build simulations of the normal tubular structure in the kidney, change the adhesion properties of the cells. The adhesion change leads to changes in the signaling that regulates cell proliferation. So in normal tissue, when the cells are touching each other appropriately, the cells have a signal that says the tissue density is sufficient, I don't need to divide. When there are gaps in the tissue, that signal disappears, and the cells at the edge of the gap will proliferate to fill the hole. So this is a normal tissue repair mechanism. In this case, the change in adhesion changes that signal and leads to inappropriate proliferation of the cells. And that inappropriate proliferation continues, leading to the formation of cysts. Bob did some nice experiments in vitro where he took cells from a human kidney cell line that normally form tubes, put in the specific cell adhesion molecule that he believed was the problem in this disease, and showed that that led to the formation of cysts. This green color means that those cells have the extra adhesion molecule that wouldn't normally be present. And you see spherical cysts wherever that molecule is present. We then worked again with a group in Brazil, Rita de Almeida, also at UFERGS in Porto Alegre, doing some classic bioinformatics analysis to try to understand whether this particular defect could be remedied with a drug. And we're actually now trying to do clinical trials. We're not at clinical trials, we're still in animals, of potential drugs to try to remediate this problem. We've worked a lot over the years on liver toxicity. This may not seem to be very connected. One of the problems that you face in both chemicals that are used as drugs and molecules that are toxic is that the first place that things come from when they are absorbed in your intestines or your gut is the liver. And the liver does a lot of work to try to eliminate extraneous chemicals from your blood, detoxification. In the process of that, sometimes it takes a molecule that's not very toxic and makes it more toxic. Ethanol, alcohol is a classic example of that. Another one of those is acetaminophen, sometimes called paracetamol. The technical name for it is APAP, APAP. That's a headache remedy, fever remedy. It is itself not very toxic, but in your liver, certain cells in your liver turn it into a molecule called NAPQI, N-A-P-Q-I. That molecule binds to a buffer in the cells called glutathione. And when the cell's glutathione level goes too low, the cell dies. This is a model where we've taken a large-scale model of the transport of these molecules in the whole body. We've opened up the liver compartment. We've built an explicit model of flow in one of the sinusoidal vessels in the liver. And we've modeled how that particular molecule gets into the cells, is converted to these toxic products, and then comes out, and then validated it against clinical measurements for that. That kind of measurement allows you to ask the question, is a person at risk of getting sick from this? A lot of people die every year from acetaminophen toxicity. And you can then do classic sensitivity analysis. You're not going to be able to get much out of this picture. These are a bunch of measures on the vertical axis of different metrics of how damaging the outcome is. On the right-hand side, we have the many, many parameters in this model, and we can identify which of these parameters are the ones that cause the most problem. That allows us then to go back and build something that's very useful medically, which is a simulated population. So what we do is we fit the model to the clinical data to get a typical patient. And then we say, now, if we allow all of our parameters to vary by a little bit, we can generate a family of possible virtual patients and ask, how do they respond to this drug? The answer is that on average, that's the black line, they respond the way the average patient responds. And the variance is the same as actually seen in the clinical population. But even though none of the parameters individually are changing very much, there could be reinforcement. So some individuals have very low response to the drug, so they're protected against toxicity. But a few unlucky individuals in the simulated population have a very high sensitivity to the drug. And this is seen in many drugs where most people are fine with it, but a few people are unlucky and are very sensitive and can be poisoned by it. And you might expect, like in the case of polycystic kidney disease, there was a specific gene mutation that caused the problem. In this case, every gene is within a normal range of variation. It happens that you have a combination of changes that are more than additive that cause a problem. Since we're a physics audience, I wanted to show some flow pictures. This is actually work that we did collaboratively with our School of Medicine. This is actually looking at flow in the liver of a mouse as it's alive, and you can see the flow. One of the things that's rather interesting about the flow is it's not uniform. It stops and it starts, it even reverses. And so we did some nice simulations trying to understand how the flow pattern in a realistic model of the geometry of the liver changes what's called microdosimetry, what level of toxin you're exposed to in a given place in the liver, what level of oxygen you have in the liver, and so on. The last thing I wanted to talk about today was more recent work, talking about infectious diseases. There's so many things we still don't understand about how viruses spread in the body and about how our immune response works. There's also been a lot of work on antivirals. We know about drugs like remdesivir. There have been a lot of failed antivirals. And so one would like to understand how to optimize treatments, both things like antivirals and also immune modulators like dexamethasone. We're going to build a very simple model of a patch of lung tissue. The actual structure of the lung and what's happening is very complex. We'd have to understand really a lot to build a detailed model. We're going to start very, very simple. And we're going to imagine we have a sheet of cells, which are all the same to begin with, that are the lung cells that can be infected. We're going to model immune cells that can move outside of that tissue. And we're going to model a viral load where the virus is modeled as a chemical field, which can spread and infect new cells. And the individual cells then can produce virus themselves once they're infected. We're not going to deal with the complexity of the adaptive immune system to begin with. We're going to assume we're dealing with what's called acute primary infection. Your body's seeing a new infection for the first time over a relatively short time scale. We can take this complex biological idea, simplify it into a couple of cell types, epithelial cells, immune cells, field, which represents viral density, a field that represents the cytokines, the signaling molecules the immune system uses. And also there is an oxidative chemical field, which the immune cells secrete to kill cells in their environment. Once a cell in your body has a virus inside of it, the only way it's ever going to be dealt with is to die. Your body in general doesn't have the ability to shut down. There's some exceptions always in biology, but in general, your body can't shut down viruses. Once a cell is infected, the only thing your immune system can do is kill that cell. This is a simulation here showing an initially infected cell. Other cells become infected, release more virus, eventually all of the cells die. Here is a layer of immune cells that are coming in and trying to prevent the spread of the virus. And you can look at the viral chemical field, the chemical signaling field, oxidative agent. If we want to ask the question, how does the virus reproduce? Each one of these cells has a model of viral reproduction running inside of it. We can then imagine that we are treating the cells. In this case, where remdesivir blocks the exponential replication step of the viral genome. And we can then simulate what drug treatment does as a function of concentration. Towards the top is a higher dose of the drug and the time when you start. Because one problem with these viruses is that you don't start feeling sick until you've been infected for quite a while. And so you don't start treatment until several days into the infection. Green here means that the treatment was successful. Red means the treatment was unsuccessful. White means that sometimes it was successful and sometimes it wasn't. And you can begin to build phase diagrams of how this treatment works. More recently, we've been trying to do this in a little bit more detail. In that simulation, once the treatment was turned on, it stayed on. In reality, you give the drug, it takes a while to get into the cells. In particular, remdesivir is not active, it's the metabolite. In the case of APAP, acetaminophen, the metabolite was toxic. In this case, it's the metabolite that's doing the work. That means it takes a little bit more time. The drug then is eliminated from the body between doses. And so you have this sawtooth where you apply the drug, builds up, it goes away, it builds up, it goes away. In these periods where the drug dosing is low, potentially the virus can replicate. And so you then have to adjust the dose to make sure that the virus isn't replicating in the gaps between treatments. In this case, we're saying treating every day, every 2 days, once a week, and so on, longer dosing periods, and asking the question, how does the drug work? Again, red means that your drug isn't working. Green means the drug is working. Blue and black are intermediate states. One thing that was modeled here was that not all cells are the same. Some cells might take the drug up faster or destroy the drug more rapidly. If you include that variation, the drug is less effective, which means to get the same output, you have to give a higher dose. That model didn't include all of the complexity of the immune system. This was a model which was done with Bard Ermentrout and Erica Mohan in Pittsburgh, which took a very detailed and well-validated model of influenza infection in mice, where they had extensive experimental data on all of these different immune components, and actually built a model where we had macrophages, natural killer cells, CD8 plus T cells, CD4 plus T cells, B cells, antigen-presenting cells, and actually tried to model the complexity, the real complexity of the immune system in a tissue, and able to reproduce again this kind of clinical output. With Giulio Belmonte, whose name you've heard many times already, we've also worked on trying to use this kind of approach to do whole lung modeling. That's just getting started, but is something that might be interesting for people to think about. They took CT, micro CT images of the lung and built structural models of the lung, and then looked at how the virus could spread within the lung. Another thing that we've been working on is trying to go beyond this lattice-based approach to modeling. We've been developing a new software package called Mechanica that's more, it's off lattice. It's based on molecular dynamics code. And we're just beginning to release that now. And it has some interesting properties and features, if you're a physicist, for building this kind of model. So I'd like to end by just saying that we like to always look for collaborations. If you're interested in modeling, we work with people who help model cancer, blood vessels, developmental phenomena, toxicity, bacterial colonies, and so on. The models that I've shown are extensible. They're designed to be frameworks that people can use to build their own models. In particular, these viral models are quite complex. And so if you want to switch it from influenza to modeling hepatitis, that can be done, and we can help with that. We'd love to work with people doing experiments or other kinds of work on trying to make these models more realistic. If you're interested, particularly in infectious diseases, as was mentioned earlier, I'm a co-lead of a National Institutes of Health working group discussing these things. In fact, normally I'd be right now leading that discussion. It's on Thursdays at 3 p.m. Eastern Daylight Time, so 2 p.m. for you. If you're interested, you are welcome to join. You can sign up, attend our meetings if you like, no obligation, and learn about modeling in viruses. We have different people every week talking about it. For physicists, it might also be interesting to talk about extending the computational architecture. Computational is a modular framework. Each one of the descriptions of a biological process has the form of an energy function. And so if you have an idea for a curvature energy term or a long-range interaction term, and so on, we can help you add that to Computational. It's open source shared software. We'd love to have people add new object classes. Maybe you want to model particular kinds of fibers or particular kinds of materials. If you're interested in solving reaction diffusion equation or advection diffusion equation, we'd love to have that. If you're interested in GPUs or parallelization, we'd love to talk to you. Computational is written in Python. It's written in C++ with Python interfaces. So if you have code that solves fluids problems or solids problems or radiation problems, and it has a Python interface that can be coupled to Computational, we're very interested in doing that. We're working on artificial intelligence or machine learning methods to speed things up, to find parameters, to do optimization, so on. And so those are all possibilities, and we're interested in a whole workflow how do you go from experimental images or medical data to simulations and back. So anybody interested in any of these problems, we'd love to talk to you. I hope that gives you an idea of the kinds of things you can do, starting with the Ising model and soap bubbles, and about how molecular and mechanical signals work together to build tissues, maintain them, and occasionally to fail. Again, if you're interested in playing with CompuCell, you can download it from cc3d.org. It's also available online, as I say, if you want to run it without installing it on your computer. At the beginning of August, we taught our annual CompuCell course. That's a week-long course in how to use it. All of those lectures are online, so if you want to watch them, it's maybe a little intimidating for a single sitting, 30 hours of lectures, but they're available. And we'd be happy to work with you if you have any ideas for what to do or how to have problems using it. And you can always contact me, jglazier@gmail.com. Current and recent funding sources in the United States. And since I talked about remdesivir, I have to say that as part of my pension plan, I own a little bit of Gilead stock, and Gilead is the company that makes remdesivir. I don't have anything to do with the company except for the fact that I own stock. But I have to disclose that. Thank you very much for listening.
Well, thank you very much. Very nice talk. So I think, are there any questions? There's some questions. Let me, please, Alejandro. Go on. Alejandro, can you hear me?
Yeah, I can hear you, but I don't have a question, only my congratulations to the speaker.
Okay. So are there any questions from YouTube? Because let me see. Let me ask. Alberto, ¿no hay preguntas por el YouTube?
Rather, okay. I have a question.
Okay, go on, please.
Thank you very much for the conference and the presentation. And I have a question about aging. How you can model the reduction of functionality of some parts of the system?
Well, aging is, of course, something that is still very poorly understood and very complicated, because aging happens at multiple scales. Individual cells change their behavior. The fraction of cells that are stem cell-like and the behavior of the stem cells that renew tissues change as we age. And many extracellular structures change as well. And so aging isn't a single problem, it's a problem that occurs in a variety of places. You have damage to DNA that accumulates due to reactive oxygen species, for example. You have damage to mitochondria that accumulates because the mitochondria aren't renewed in the same way that other cells are. And so those are things that are happening inside single cells. There also are tissues, most tissues in your body are renewed. Even things like bone are renewed over a period of 15, 20 years. The bone is remodeled. But there also are certain things that are laid down during development that are not renewed. The classic example of that is neurons, where most neurons, once the neuron has formed, if it's destroyed, you don't replace it. With muscle, certain muscle tissue, if you lose myofibers, the cells forming the muscle, you don't make new muscle fibers. The ones that survive get bigger. In what's called cancellous bone, the foam-like bone in the middle of long bones, like the tibia, there are fine-grained bone fibers, which are not replaced if they break. The rest of the bone gets thicker to compensate for the loss of the fibers, but the mechanical properties change, and the resistance to fracture goes down. Even if the total amount of bone doesn't change, the resistance to damage goes down. Another place that happens is in major blood vessels. Some of the extracellular fibers that give the elasticity to blood vessels, some of those are renewed continually through your life, but there are certain ones of those fibers that are laid down when the vessel forms and are not replaced. And one of the reasons that diabetics have blood vessel problems is that the sugars that accumulate in diabetes break down some of those fibers that surround the blood vessel and effectively cause premature aging of the blood vessel. Another place that this happens is in cartilage. Used to be believed cartilage didn't renew itself at all. It's not true. It has some renewal capability, but not a lot. And so damage to cartilage is typically progressive. As you age, the extracellular cushioning that cartilage provides can be lost. And so you have degeneration that way. So there are many different aspects to aging. All of the things that I described could be modeled using the approaches that I've described, but you would have to build a separate model for each one of those problems. I don't think there's a single universal approach.
Thank you.
So that was a very long answer, but I think it was a profound question, so I wanted to take it seriously.
Thank you very much.
Are there any more questions? Maybe in YouTube? While the people ask some questions, I would like to ask you just a very simple question. Two questions, in fact. The first is that you started with the Eisen model, then you move to the POTS model, which you have more components. So I was wondering why you move and how do you decide which POTS model? That's my first question. Yeah.
Well, how did I get involved with the POTS model? I was working on foams, so fluid foams. And there was a group at Exxon Research, Gary Grest, who's now at Sandia National Laboratory, Srolovitz and Anderson, who were doing material science applications. And we realized that the material science that they were working on, POTS model, could be applied to foams. And so I spent some time working with them on foams. And then I had decided already as a graduate student that I wanted to begin to work in biology rather than purely in physics. And so I moved to Japan. I postdoced in Japan. And they were interested in this problem of cell sorting, of this reorganization. And I was doing experiments at that time primarily. And I realized that this foam model that I'd been developing with Exxon Research could very easily be generalized to describe the movement of those cells, that cell reorganization, cell sorting picture that I showed. And so it actually only took a few hours to take the code that I had written originally to simulate soap bubbles and turned it into a simulation of cell reorganization. I never thought back in 1991 or 92 that 30 years later, I'd still be using the same methods. I assumed at the time that you'd get some small insights into the biophysics and you'd move on. But in fact, because of this pseudo-Hamiltonian structure where you can add additional biological mechanisms by adding energy functionals, the method is extremely generalizable. And so in a sense, I always thought it would break and then we'd move on. But so far it hasn't broken. Maybe that's a failure. Maybe it would have been better if it broke and had to start something new.
Yeah. And my second question is there is a kind of dictionary when you have certain defined, predefined. So, because to me, it seems that you need to put each time that you put some functional, you put some new parameters in the model. So is there some kind of universal dictionary or something? Or?
Well, there should be. That's probably a defect of the modeling framework, that we don't really give you predefined parameters. There are certain kinds of rules of thumb. There's some common sense ideas. So for example, cell-cell adhesion is very important.
Mm-hmm.
But as we know in physics, if you know that whether you are ferromagnetic, or antiferromagnetic depends basically on the sign of the interaction, not on the magnitude. And so what you find very often is that the exact values of parameters are not so important, but the hierarchy, whether one parameter is bigger than another, is very important, because that controls which phase, part of the phase space you're in. But the absolute value may not matter so much. Okay. And so you can then come up with rules about this needs to be bigger than this must be bigger than half of this, and these kinds of rules. Those rules exist. And then you need to go back to the experiment to say what the exact values are. But the qualitative behaviors typically depend on the order of the energy terms, the relative values, and not so much on the exact values. And that makes it much easier to develop.
Okay, thanks. So maybe we have time for one more question. Okay, if not, the director of the institute, she needed to leave, but she thanks a lot you again. And thanks again for being here. It was really a very, very wonderful talk. Thank you very much.
Thank you. And I hope if anyone is interested in any of these problems, please contact us. We're always looking for opportunities to come help people do the work you want to do, or collaborate more intensively on projects. So please,
yeah, we have a whole part of the institute that works in medical things applied, physics applied to medicine. So I think there should be a lot of people interested in this kind of work.
Okay, well, thank you very much for spending an hour with me today. I appreciate it.
Thank you very much for giving this talk.
Thank you. Goodbye.
See you. Bye-bye.
Goodbye.
Thanks to everyone.